what do you have to read into a question? Okay, what do you have to read into a question? So, when we look at the first question, it is always important to check what information is provided. The stages in sexual reproduction in a mammal. And from organ P, and a cell from the testis. They will go through a process, and then together they form a fertilized egg and a process, and now there's an embryo, and process Z will form lots of different types of cells. What is the name of organ P? Organ P, ovaries, because it was an egg cell or an ovum. Give the names of parts A, B, and C in cell 4. Nucleus, cell surface membrane, cytoplasm. What is the name of cell 3? It is a sperm cell. What type of cell division takes place in process Y and Z? Mitosis. Because it is one cell that increases in number due to cell division. What was the name of process X, if you care to go back? X was to form a gamete, so that was meiosis, right? Now it was mitosis. Which two of the cells labeled 1 to 6 are genetically identical? And that will be after the ovum and sperm fused, fertilization took place and a zygote formed. That zygote then start to divide into an embryo. So the zygote and the embryo, 5 and 6, definitely was genetically identical. Good. The gametes, we know, was the sperm and the egg, so it was 3 and 4. The cell 6 change into a range of different cell types. The name for these different cell types was stem cells. And when they change into many different kinds of cells, it is due to differentiation. Because the structure of the cell changed for a special function, the cell differentiate to have a special function for specialization. Differentiation is what we can see happen to the cell. Specialization indicates the new functions that this cell can fulfill. Cystic fibrosis is a condition in which people suffers from the accumulation of thick and sticky mucus in their lungs. Cystic fibrosis. Now, here we see males and females that suffers from this, and I want us now to have a clear understanding of the female and another female. Those that are colored is unaffected. Those that's only a circle, it has cystic fibrosis. So here we see a female Trudy, so all the circles are female, and Sadia, they are unaffected, right? Angela and Kirsty are affected females. The male, when it's a square, we have the cystic fibrosis males, and then when it is a filled square, they are unaffected. So we see that Liam is unaffected, Farad is unaffected, Josh is unaffected. Right. Gareth also unaffected. But Richard is affected. Good. So, use 2 pieces of evidence from the family tree explaining why cystic fibrosis appears to be a recessive gene. A recessive gene. So recessive means it is a gene that when the gene is paired with another recessive gene, we will see the disease, the phenotype of the person will be cystic fibrosis. When it has the dominant, not affected gene, that person will not be ill. You agree? So, when we look at Angela, we will see Angela has the disease. So Angela is FF, right? And Farad is big F, small f. Liam is unaffected. But Liam must have an F. Angela inherited a gene from her mother, which was a small f, but from her father also a small f, although he is not affected. So it is telling me that although that dad is not affected, he contains the gene. Now, and that is how I know if he can have a child with the disease, he contributed to the genes of Angela. And that then therefore he was not affected. So his small f, his recessive gene was masked off by his not affected gene. And therefore it must be recessive. Josh and Sadia is not affected. But they both inherited the gene. So they inherited the F gene, but they are not affected, so I know that it must be a recessive gene, because you can see they are not affected. Okay, right, so Trudy and Gareth want to have another child. What is the chance that this child will inherit cystic fibrosis? Trudy and Gareth. Trudy and Gareth is unaffected, but they have already 2 children that are affected. So, if we look at the phenotype of Trudy and Gareth, they are heterozygous not affected and crossing with heterozygous not affected. The phenotype of the parents looks like that, and when we look at the gametes, and please remember, we always circle gametes, we will see that there is the possibilities. And just to make it a bit easier to see the different fertilization, we see that we have offspring 1, 2, 3 that is not affected and 1 that is affected. So we will have normal to cystic fibrosis 3, 2, 1. So is there a possibility? Yes, they always have a chance of 1 out of 4, or a quarter chance, or 25%, or a ratio of 1 to 3 for cystic fibrosis to normal. Take care that you indicate in which order you do give your ratio, please. Okay, the next then, who had the different combinations? Liam had a dominant and a recessive allele, Angela recessive, and Sadia also B dominant and recessive. Explain how it is possible that Farad and Angela could have a child with cystic fibrosis. Again, we said Farad is heterozygous, so Farad looks like this. Angela looks like this. If a kid inherit from Farad's sperm that F, from Angela, the only possibility will be to inherit a recessive F. Their children will have a possibility. And remember, when we have heterozygous, homozygous, then there will always be a 50-50 percentage, a 1-to-1% of having a cystic fibrosis or an uninfected child. Okay, let's look at the next question. This question is about the hereditary material of the human species. How many chromosomes are found in the human karyotype? There are 46. They are 46. How will the chromosome composition of a human be in a somatic cell? In a somatic cell of a female, it will be 44 plus XX. In the male, it will be 44 plus XY. In the gamete, it will look like this. 22 plus X is one of the male gamete, or 22 plus Y. So a male gamete has two options to divide. Just to conclude, the female gamete will look like this. 22 plus X. 22 plus X. Okay. Hemophilia is due to a sex-linked gene. Hemophilia is that disease that we talked about in the video of the royal family, where when you have a heat against your body, you can have internal bleeding, or it's very easily for you to bleed because your blood does not clot or coagulate very easily. So, the question is, hemophilia is due to a sex-linked gene. It is recessive and found on the chromosome. A woman who is on the X chromosome, it is on the X chromosome. A woman who is a carrier for hemophilia marries a normal man. What will be the possibility phenotypes of their children? Use the letter H and use a Punnett Square with labels. So, if it is a recessive gene, the gene for hemophilia looks like this. You agree? So, if we are going to put in a Punnett Square, let's just move down our Punnett Square. I'm just going to clear here so that we can easily see the correct answers there. Okay, so we say we have a normal man, and it is a sex So the H is the gene for hemophilia, right? A normal man will have an X and a Y. Because it is only carried on the X, there will be a capital H. There will be nothing on the Y. That is the male. Okay, you agree? Male. So let's write down the male in there. And the female is a carrier. So she will have the normal and the recessive. Because we are talking about gametes, we need to put in our circles. Right? We need to state that this is the male and this is the female. Right? And we need to state that this is meiosis that makes this possible, please. Okay. Good. Now we are going to look at fertilization. And here we will have X, H, X, H, X, H, and X, Y. We will have X, capital H, X, H, and X, H, and Y. Okay. What will the possibility phenotype of the, what will be the possible phenotypes of their children? So their children will have their, we have a homozygous normal child. And what will it be, male or female? It will be a female, no? It will be a female. Right. This one will be a heterozygous. And when you hear the word heterozygous, in the case of a recessive disease, we actually know that it is a carrier. A carrier for hemophilia, and it will be a female. Right? These two are covered in there and there. Now, this will be a male, and that male will be a normal or normal male, right? And this male will be a hemophilic male. Okay, I hope that you now with sex link we see not only who's got the disease and who does not. We also see male and female in this answer. Now, okay, right. A phenotypical normal man who has a hemophilic brother marries a normal woman who is not a carrier. What is the probability that any of their children will be hemophilic? Now, a man can only be either a sick hemophilic or a healthy person. You agree? So this man is normal, and his wife is normal. Yes. I had to use a H for hemophilic. Let's do this again. Going to try and get us that very thin line now. Okay, so a male looks like this. H for normal and a Y. The female is normal and she's not a carrier. She's H and H. They are as normal as can be. So what is the probability? They will have 0% chance to have a child with that. Okay. Color blindness is an X-linked recessive trait. If a normal-sighted woman whose father was colorblind, what is the probability that they will have a son who is colorblind? Here's the letter B. So, colorblind is a recessive trait. So we will have a small b, right? A normal-sighted woman will look like this. On the X, she will look like this. Because this X of this normal woman came from her father. So this woman is definitely a carrier, unfortunately, for her children. She's definitely a carrier, nah? Because her father was colorblind. One X she got from her mother, but the other X came from the father. The father had only one X. So it was then inherited, not given. That's wrong to say it was given to, because it's not giving. He makes his children inherit his genetic profile. Now, so now she married a colorblind man. So we have to start with the phenotype, colorblind father, crossing with a normal phenotype, the genotype, small b. The gametes will look, and we have fertilization, and then we have the offspring, genotype, and we have the offspring, phenotype. Good. So the fertilization will look like this. X and a X, B and a B. Immediately, I am trying to tell myself that is a female. Right. Be careful. Then the next one, that and that is an X, B, X, B, and that is again a female. Then I have a Y, so that will be a X, B, and a Y, and there will be a X, B, and a Y, which will be the males. So what is the probability that they will have a son who is colorblind? They have a possibility of 2 sons, right? You agree? And now there's a 1 out of 1. There's a 50% that one of the sons will be color. Let's continue. In man, muscular dystrophy is a condition in which the muscles waste away during early life and may result in a shorter lifespan. It is due to a sex-linked recessive gene. A certain couple has 5 children, 3 boys and 2 girls. The boys are 1, 3, and 10, and the girls are 5 and 7. The oldest boy shows symptoms of the disease, but none of the parents shows any symptoms. If the boy is showing symptoms and it is a recessive, it means that it is a sex-linked, so it means, if we use a small m for muscular dystrophy, the boy will look like that. And the boy can only inherit this X from whom? From his mother. Because remember, the mother, the mum, is this. You agree? The boy do have the symptoms. So, he must have inherited this from his mum. What about the dad? The dad is X, Y. Right? But that, if this boy, the only possibility for him to got this disease was from his mother. Now what's the question? You are the family physician and they come to you for advice. What would you tell them about the chances of the other children developing this disease? So, now we will need to make a Punnett Square or something in order to help us to sort this problem out. So our Punnett Square, let's insert a Punnett Square here. The mother, the father, this, because he does not have the gametes. Here, this girl will be homozygous normal. Right? This will be a boy, and this boy will be homozygous, it will be normal. Now, here we will have a girl again, 2x, but she will have a capital M, small m. Capital M, no disease. X, capital M, no disease. Small m, capital M, this will be a carrier girl. And the last one will be a boy again with a small m. This will be a boy with the disease. You see, so when they have a 1 out of 4 chance, 1 out of 3, 1, 2, 3 chance to have a ill child with muscular dystrophy. And it will be the boy. Therefore, class, they normally says that it is the mum that carries the disease to her son. If the mother is a carrier and the dad is healthy, it's only the X on the mum that will be the gamete that's carrying it to the boy. I hope that you do understand that. What is meant by sex-linked genes? And this is now on page 501. What is meant by sex-linked genes and sex-linked disorders? A sex-linked gene, a gene found only on sex chromosomes. So we saw in these questions hemophilia, color blindness, and muscular dystrophy. These are all diseases that is a sex-linked disease because it is attached or on the X chromosome. Male do not have 2 X's but only 1 X. So if a male inherits the dominant gene, he will have the condition. If he inherited the recessive, on the Y there is nothing to mask it off. He will be healthy. If a female has one of the dominant and one of the recessive, she's a carrier. Okay, right. Any disorder that is determined by the sex chromosome or by a defective gene on a sex chromosome is a sex-linked disorder. Then one last question, help us think. A man with normal color sight marries a woman who is carrier for color blindness. What is the probability for their children in percentage to be color blind? So let's write in the male. Going to put the male here. So the male looks like this. And the female, the male X, N is the one gamete for the male, and Y is the other gamete for the male. The female X, N, X, small n. She is the carrier. So this child will be a X and a X, so it's a girl. Capital N, capital N. So normal vision. N, another girl. So these two here we will have girls, right? There we will have now an X, N, and a Y. X, small n, and a Y. And this will be the boys. Now, there's always a 50-50 percentage to have a boy or a girl, because we have a heterozygous and a homozygous crossing, always 50-50%, right, or 1-to-1. But to have colorblind children is 1 out of 4 is 25%. Our answer there will be 25%. The next question, which we are also going to use this, how many daughters will be normal? 1 out of 2. So 50%. Take care. 50% of the daughters. This was not about children. And if 50% is normal, in this case, we will have 50% carriers. What if a daughter marries a colorblind man? What if the daughter marries a colorblind man? And I just want to make sure is a carrier. So the daughter who is a carrier marries a colorblind man. Now we have exactly the same scenario. We have the daughter who is a carrier. Please remember this is gametes and circle them. Marries a colorblind man. So small n and a y. Now, they will have a carrier daughter and they will have a colorblind. They will have a possibility to have a colorblind daughter. And their sons, they may have a normal son, but they also may have a colorblind. So they have a 50% chance of having a colorblind child. Now, they have a 50% chance of having a colorblind child, because 2 out of the 4 children, that gives me 50%. Okay, right. And the last question is, Color blindness is more common in men, as I have explained, because it is carried on the X. Man has only one X chromosome, and therefore there is not another one to mask off. Okay, grade 12, please make sure that you do understand this work through all the different possibilities of how inheritance problems can be asked. DNA stands for deoxyribonucleic acid. Where is most DNA found? In the cell of a eukaryotic, and that is the nucleus. A prokaryotic is in the cytoplasm. And the red blood cell has no DNA. State the names of the bases. Please spell correct. Or so that when I say your name, that it is correct. But higher level, they are strict on specific scientific words that you have to spell it correct. Okay, so if one must be thymine, because thymine links with adenine, and guanine will link to cytosine. The component X, I want to see deoxyribose, because you are higher level. It's not only a sugar, it has a specific name. And then, of course, Y, the phosphate group. Thank you. Many of you remember that. Purine is guanine and adenine. And I realize that a way to remember, the pyrimidines are the smaller ones, the ones that is only one ring. Yes, somebody said the purines are 2 rings. Nah, the pyrimidines is only 1 ring, but we can also remember thymine has a Y and cytosine has a Y. And that will then help us to remember pyrimidine, thymine, cytosine. Now, that is also a way to remember those 2 different ones. Then, describe why, the first question there was, diagram shows a process taking place and will change the DNA diagram. The process visible is replication. There stand 1. Why is it important for this process to take place? We need identical copies of the genetic instructions to be passed on to a daughter cell. So during cell division, if you say during cell division, identical copies must be made for the daughter cells. That is also a good answer. Then here, for 6 marks, I think that it is important to say the DNA will unwind and unzip. This happens by helicase, and I thought that earns a mark. Break hydrogen bonds between the nitrogenous pairs. That you cannot just say the hydrogen bonds break, because unfortunately there are hydrogen bonds between other parts, between inside the cytosine to make that cytosine circle. They can be hydrogen bonds. So the hydrogen bonds break between the complementary nitrogenous pairs or between the nitrogenous pairs. I need to know where the hydrogen bond is that will break. Allow the strands to separate. Bases are exposed. There we can see the bases are now exposed for a nitrogenous base to attach. Now, bases are exposed for the free nucleotides with its nitrogenous base to attach. Complementary base pairs happens at A to C and C to G, and ligase help to form the backbone. DNA polymerase can be in the next line. Polymerase, DNA polymerase links the nucleotides to identical DNA molecules will form. So make a simple drawing, and what I think was important for that drawing is that you have now strand 1. Now, so here we have strand 1. There was strand 1. So you draw there and you show me the nitrogenous bases. But that is not enough. I need to see how are they going to attach. So A will attach, and this is DNA replication. So this is another DNA strand. And A will link to T. Yes? So that will be a blue one. There is another A, T. And T will link to A. That one will be a red one. And then G and C. So let's just make C. Let's make C green. C. That one looks like, and there's, okay. So it will link to a yellow, and G will be green. You agree? But you cannot use color, so let's rather write in there A linked to T, T to A, C to G, G, T to A, G is yellow, so that's a C, C, G, and T. That is how the other side will look. Make a simple drawing to show the end of this process for strand 1. So there I just link them. So the end of the process will look like this. 8, and I will have A, T, T, A, C, G, C, G, T, A, G, C, C, G, A, and T. And this is the end of the process. So for your 2 marks, I would have checked, do we have them all linked? Do I have this ladder diagram and are all the bases correctly linked. Describe why this is called a why this process is called semi-conservative. Then the answer there will be the words that one original strand serve as a template for the new strand, and the new strand, that will be my second mark in yellow, new strand to keep the original, to use as a template for the original code. What does it mean that the two strands are complementary? A will link to T, G will link to C with a hydrogen bond. Then we have the diagram. So if this is the DNA, the mRNA will be linked with a complement, right? Between those two, it will link with a complement. That means the T of the DNA cannot link with a T. That is not complementary. They cannot link. They do not fit into one another. So the T will become U in the RNA. Yes, in DNA. In DNA, T links to A. T links to A. A links to T if we talk DNA. But if we talk RNA, we know there are no T's in RNA. The T is substituted with a U. Aha. So our answer is A-U-A, right? A-U-T-A. The codon then on the tRNA will then be U-A-U. Okay, just check. So mRNA is the complement of the DNA. The complement of DNA in RNA language will not have a T in it. Indicate the location in the cell where transcription is in the nucleus. Transcription is in the nucleus. Translation in the cytoplasm, or if you say ribosomes, and mRNA is produced, but in the nucleus. Good. Use the diagram above and explain the terms transcription, the process of constructing a messenger RNA molecule using a DNA molecule as a template with resulting transfer of genetic information to the messenger RNA. Translation, the process of forming a protein molecule at a ribosomal site of protein synthesis from information containing in the messenger RNA. So I want to see the word protein molecule, a ribosome, and messenger to give you all your marks. Now compare the differences. And there on my slide you will see the differences. DNA is a double strand while RNA is a single strand. But I ask in a table, nee. So you have to show me your answer in a table, please. If the question was not in a table, you see you cannot say DNA is double. And then I have to think what RNA is then? Triple? What? RNA is a single strand. Nee. So that I want in a table, please. DNA has T, thymine, while RNA has U. DNA has deoxyribose sugar, while RNA has ribose sugar. RNA is also found in the cytoplasm as well as in the nucleus. Well, DNA is only in the nucleus. That's 4 different ones. You have more. What are the similarities? Okay, let's study. Both are nucleic acids. They are made out of nucleotides. Both have a sugar phosphate backbone, and are found in the nucleus. So there are also 4 similarities that you can quickly remember. So, thank you, great.
G forces. What are those 4 going through at the moment?
You know, they're obviously the G forces. They're going to be feeling as if they're pushed back in their seat. It shouldn't be too high. The rocket's not going incredibly fast or as fast as you would need to escape Earth's gravity and enter orbit. So I think it's probably fairly manageable. They've been acclimatized to what to expect with those couple of days of training. I think for them, it's probably just a very exciting moment to know that you're just seconds away from being able to feel just a little bit of low gravity and be able to float around the capsule and see the curvature of Earth. So I'm quite jealous of them at the moment.
I bet you are, as we continue to see the different images now being beamed live from New Shepard. Now, of course, they get to about 60 miles up, just beyond the Kármán line. For those watching who are not familiar with that, just give us a brief description of where space starts and Earth's gravitational ring actually ends.
It's actually quite a complicated question about where space actually starts. Some people say it's 50 miles up. Some people put the Kármán line at about 60 to 66 miles up. It's a very difficult question because it's about where there's a lack of atmosphere, where sort of your normal aerodynamics don't work anymore, and you can't rely on that as if you're, have wings on an airplane. So it's actually, and the atmosphere fades out fairly gradually. So this is a very complicated question. There is no sort of legal definition. Different agencies recognize it at different levels. In the United States, NASA recognizes it at 50 miles. So the Kármán line is obviously higher. They are going quite high. So it's a very technical and complicated question, and I'm afraid I don't have a specific answer, but it's sort of where there's a lack of atmosphere, where you can't rely on a normal aerodynamic flight.
Wendy, we're looking at the pictures. We just got a glimpse of the curvature of Earth. I'll let you have a look at those pictures and I'll come back to you in a moment or two. But I'm going to go to Van Horn in Texas, the launch site, because Sophie Long has been there throughout the course of the last couple of days. Well, Sophie, we've had a couple of delays, but we were watching, you were watching, we were watching. The blast seemed to go off perfectly in the last few moments.
It did indeed, Matthew, yes, and we have had it confirmed that the separation of the rocket from the capsule has taken place successfully as well. So any moment now, if not already, William Shatner and the other 3 passengers on board the New Shepard capsule will be experiencing that weightlessness, that zero gravity that they get as they pass the Kármán line, that internationally recognized border with space. So they'll be experiencing that for just over 3 minutes, and that's what people are really looking forward to. William Shatner has spoken in the last few days about how he's waiting to be entranced by that view of Earth, and the few that have boldly gone there before him have said it is a life-changing experience. Looking back at planet Earth where we are now, Matthew, and just seeing how fragile it is and how small it is. So they should be experiencing that now. After that, they will free fall back to Earth. And remember, there's no pilot on board this capsule or the rocket. It's fully automated. So those 4 passengers will then free fall back to the Earth's surface. And as they approach it, a few thousand feet up, 3 large parachutes will be deployed to slow it down. Thrusters will come from the base, so it will make a safe impact with the Earth's surface. And William Shatner, more than 50 years after he first donned a spacesuit, of course, as Captain Kirk, will now be a real-life space explorer.
use the phrase space explorer because I was listening to you earlier about the description, the technicalities around astronaut, also that whole notion of space tourism, Sophie, because of course we've seen Bezos and we've seen Branson make this trip in the last few weeks. It is really gathering pace, isn't it?
It is, Matthew. Yeah, we're just watching. I don't know what pictures you're seeing, but we're just watching the rocket coming back down to the Earth's surface. It's going to land just a couple of miles from where I'm standing here. That does not, of course, carry the passengers. They're still up in the capsule and will follow. But yes, the debate about whether they will return to Earth astronauts or not is one that is raging. You heard that sonic boom probably there, Matthew. That was the rocket making impact with the Earth's surface a couple of miles from where I'm standing. Yes, there's a huge debate over whether there'll be astronauts or space explorers, but certainly William Shatner. It feels quite exciting here, Matthew, the sounds that we're hearing on the ground just a couple of miles away from the launch pad. But whether you call him an astronaut or not, William Shatner will have been the oldest person to have travelled to the edge of space at the age of 90. Now, it is a remarkable feat in itself, and it's a really extraordinary day for the future of space tourism, for Blue Origin, of course, as well. This is only their second flight carrying human passengers. So at the moment, it all seems to be going successfully. The 4 passengers still, though, up there at the moment.
Sophie, as you were speaking, we were watching the rocket itself touchdown. It is extraordinary the technologies that are being used for that to be reusable, and of course we've seen that over the last few years, and that's become such a leap forward in terms of space exploration, in terms of the comparisons with NASA and the whole Apollo projects. But in terms of those 4 currently still in space, going through everything that you just described, I mean, the whole journey is pretty quick, isn't it?
It is. The whole journey, Matthew, lasts less than 11 minutes, assuming it goes according to plan. I'm just standing here on the ground and people are shouting, there they are. I can't... Yeah, we can see in the distance, Matthew, the parachutes which have been deployed and the capsule making its way back down to the Earth's surface. So that's the stage right now. It appears from where I'm standing to be moving quite slowly. But yeah, this whole journey lasting less than 11 minutes. So remarkable, really, that you can go up to the edge of space, experience the weightlessness, look back at planet Earth and be back down in less than a quarter of an hour.
Sophie, you're craning your neck, but we have the cameras absolutely trained on the capsule itself, and we can see...
You've probably got a better view than I have.
We probably have, and those huge windows that, of course, for those 4 give them such a brilliant view in terms of what they've been through over the last few minutes. But as you were describing and anticipating, the parachutes deployed and the capsule gently about to touch down in the deserts, and certainly in terms of Jeff Bezos, it took a while for the capsule door to open, but then of course they came out and spoke words to the reporters there, so we anticipate that in the coming minutes. But let's just listen in to the scenes there on the screen.
That was unlike anything they described. All right, we got less than 1,000 feet. All right. Great.